Good afternoon. Dr. Dan Guerra here from Vera Med and the Pacific Northwest Studios from which I launch these video lectures. Let's get started. First of all, I want to welcome you to another one of our lectures. What we do here is examine the published primary scientific literature, verify that literature, verify the evidence within it, by examining it and comparing it to the other literature, both foundationally and coherently, to try to come up with a clearer understanding of what current published research in biosciences, biomedical sciences in particular, can offer us in the field of biomedicine and in the general field of research science associated with biology, particularly those aspects of biology that are associated with pathology, pathophysiology, and biomedicine, biopharmaceutical research, and of course the underlying discipline is biochemistry, as we all know. So what we are then is Vera Med, and that is our website, and this is our website email that you can directly contact us when you have a research issue you would like to discuss with us and become our client. I'm Dr. Dan Guerra. As I said, there's my personal email. Contact me, please, for any content that you see on these video lectures. And also, if you have difficulty navigating in the web page, contact me directly, and I will be back with you certainly within 24 hours. This is January of 2018, brand new year. It looks to be a fantastic one for everyone I know and love, and I hope it's the same for you. Today, we're going to talk about phosphatidic. Oh, one more thing. Here's my Facebook, and any of the video lectures that you want to look at, all the backlog of them, over 30 of them, are going to be found on that Facebook page, as well as a lot of other things about, Western philosophy, and etc. At any rate, you can find them there. I'm in the process of getting them uploaded onto the web page, but go to the Facebook page now because that's where you're going to find them all. Again, contact me if you have any problems with them. Today I'm going to talk about phosphatidic acid, and the subtitle is the electrostatic power of lipid phosphomonoesters in pH, biosensing, and signal transduction in biological systems. So let's get started. Okay, first paper we're going to really be looking at is a Langmuir paper published just in 2018. In fact, it's not quite yet out. It'll be out in February. This is still mid-January. And it's all about looking at this molecule here. This is dipalmityl phosphatidic acid, or DPPA. So this is a glycerolipid. It has two palmitic acids at the 2 and 3 position of SN-glycerol-3 phosphate. At the 1 and 2. At the 3 position is just this phosphate monoester. Okay. So at any rate, what this paper is describing in biophysical terms is the protonation state of that phosphate head group, which is shown in red here. And it corresponds to, and we're looking at the level of protonation, and that would then alter the electronegativity of the molecule from -1 to -2 when you lose both protons. And that's what's shown down here. So low pH where the head group is singly deprotonated, and at high pH conditions where the head group is fully deprotonated. And those are the two different physical chemical states that we're talking about. So as it turns out, the protonation state of any organic surfactant molecule, that's a compound that's going to interact amphipathically with surfaces, particularly surfaces that have an electrostatic charge, and then usually a hydrophobic or neutral layer that is associated with that electrically charged surface. That's what a surfactant does, like detergents or surfactants. Anyway, we want to know how the protonation state of an organic surfactant like dipalmityl phosphatidic acid controls monolayer fluidity, surface organization, and membrane topology and morphology. So again, we're talking about solution pKa. pKa's are just the negative logarithm of the dissociation constant for, in this case, the protons. Okay, I'm not going to talk about the chemistry of that. Certainly, if you contact me, we can give you a primer on Ka and pKa in acid-base chemistry. At any rate, we're looking at the speciation of these particular molecules as surfactants. And what has been determined is that the solution pKa is usually not the actual pKa, because the pH and pKa of an organic surfactant is distinct at that surface compared to the bulk fluid layer. So that's what this is all about. And of course, in biology, we always have a surface, and that surface that we most talk about is the membrane surface. And of course, the membrane is composed of lipids and proteins. So by examining the molecular expansion at a basic pH, the pKa to remove the second proton from DPPA, which is called here pKa2, okay, was estimated. And this is looking at now an aqueous surface, pure water, or in an aqueous solution with sodium chloride. Now that's going to change the electrochemical potential of the fluid, and it's going to alter then the physical chemical dynamics of this alteration of electrostatic interaction with that phosphomonoester head group. Okay, so you have a condensed DPPA monolayer at a surface pKa2 of 11.5 pH units, and that surface pKa2 is further altered by the presence of sodium cations, as you might guess, because it's an electrostatic interaction. And you actually reduce it at an entire pH point, lowering that pKa, making it more acidic. It's also found that the surface pKa value of DPPA is modulated by the packing density. Now that's really important in biology. By that I mean the surface charge density. And where do we get that from? We get that from the molarity of the lipid molecules, which have electrostatic charge or surface charge, such as phosphatidic acid, which is a cone-shaped phospholipid that you find in membranes. So the surface charge density is actually the driving force behind changes to the surface pKa2. Now that's really important, as losing that extra proton, as we're going to find out, is going to allow the phosphatidic acid to interact with proteins and other phospholipids. And that's why signal transduction and pH sensing are associated with this electrostatic interaction. In fact, there's a diffuse double layer of ions consisting of counterions and co-ions, and that's embedded within that dielectric medium. And of course, the distribution of those ions is going to follow a Poisson. Okay, so that's a sort of a given, but it's also what has been physical-chemically determined. So remember where we get phosphatidic acid, what it looks like. There it is. There's palmitoyl phosphatidic acid. Okay, and here is shown carrying a charge of -2. All right. Now remember that PA can be synthesized via diacylglycerol, via diacylglycerol kinase, and DAG can be formed from triacylglycerol, or a neutral storage depot lipid. DAG can also be used to form phosphatidylethanolamin or phosphatidylcholine, and in fact, phosphatidylcholine can go through phospholipase D reaction to resynthesize phosphatidic acid. In fact, all the phospholipids, if PLD acts with them, will regenerate just the phosphomonoester. Now, PA also can swap out with CTP, that's cytidine triphosphate, to make, after removal of that gamma phosphoryl, CDP diacylglycerol, and that's actually an intermediate in the synthesis of this whole clan of phosphoglycerolipids: phosphotylserine, phosphatidylethanolamin, and phosphatidycholine. That's just methylating that choline with 3 methyl groups, methionine. Remember, too, that PA is a precursor to phosphatidylinositols, and those are really important signaling molecules in their own right, both at the level of the phospholipid and just at the level of inositol phosphate. All right? Very important, critical interactions here, all of the membrane generating an entire series of signaling glycerophospholipids, all of which can react, you see, with amino groups. So that's the point about these phospholipids. They can interact with positively charged amino groups, either in proteins, particularly basic proteins that have arginine residues or lysine residues, as well as with any free amino groups with other phospholipids. And that would include things like phosphatidycholine and phosphotidylethanolamin, even phosphotylserine. Okay, so again, you have this phosphomonoester head group that's shown in red, okay? And it's attached just basically to a DAG or diacylglycerol backbone. Now, Lipids like phosphatidic acid are involved directly with bonding to effector molecules, such as other lipids, or, as I've said, proteins. And they bind and recruit them to various membrane structures, including the plasma membrane. Okay, so just having that phosphomonoester on the phosphatidic acid will allow those lipids to bind and recruit phospholipids with a positive charge, as well as basic proteins, such as ones containing arginines and lysines. And in fact, the binding is directly controlled via membrane availability of the phospholipid, and thus it's concentration dependent. And that's what you're showing here. So you have a lipid effector protein here, and it's looking to bind to a membrane-associated lipid, such as PA. So an enzyme can come along and alter this structure. How? For example, if that's phospholipase D, it can take phosphatidylcholine, which is just your standard trimethylated ethanolamine glycerol phospholipid in the membrane, turn it into, that is, the reaction product is going to be PA, shown here. And because the PA now has that electrically charged, at -2 charge, right, because of deprotonation, because of the pH interaction, will allow it to bind to this lipid effector protein, and then therefore a signal can be transduced. Now this comes from a paper from BMC Biology in 2011. So the modification of the membrane lipid species via enzymatic turnover, such as phospholipase D, which will generate phosphatidic acid, or in fact diacylglycerol kinase, those are enzymes actually that are in the membrane, which would make PA from DAG in situ, that is in the membrane, is coupled to upstream signaling and membrane fluid dynamics. The entire membrane fluid dynamics is going to change because of this change in the monolayer, which is the outer leaflet of the bilayer. And not only that, you're going to be able to get a specific binding to proteins which may be looking to bind to surface phospholipids, such as ligands, okay? So the PA membrane concentration in general is low, only about 1% as compared to other phospholipids. But remember, it's an intermediate for the conversion to lots of other lipids, the diagram we just saw on the last slide. Plus, polyunsaturated fatty acid turnover occurs on these phospholipids, and that is all linked to signal transduction cascades such as tyrosine receptor kinases, okay? So we have receptor tyrosine kinases, which are all can be interacting with this phospholipid electrostatic molecular mechanism whereby pH biosensing associated with the loss of protons from that monoester, and then alteration of the membrane lipid dynamically, fluid dynamically, so that it can bind to ligand, thus changing the entire poise of the cell where that membrane comes from. Okay. So more from this paper from 2011, PLD will generate phosphatidic acid signaling. Okay, so remember PLD cuts right here, so you make phosphatidic acid. Here are the other phospholipases, just to give you an idea of what other enzymes can react with a phospholipid. Lots of them, right? 1, 2, 3, and 4. Phospholipase D is the one that makes phosphatidic acid. Or acts as any polar head group, or neutral head group for that matter. So PLD actually is a potent regulator of RAS signaling. This is known. Remember that RAS is a whole signal transduction cascade we talked about before that's associated with such pathologies as cancer. And it's strongly implicated because of that, PLD, not only as a biomarker, but possibly as a driver of cancer. And it's well known to regulate PLD itself, cell proliferation, differentiation, and indeed, in the right conditions, apoptosis. EGF, epidermal growth factor stimulation results in increased activity of PLD at the plasma membrane and generation of localized interactions. So the generation of PA leads to the binding and recruitment of effector protein. This is an example of what's going on here, son of sevenless or SOS, to the plasma membrane, which then leads to RAS activation. So here we're showing you the series of signal transduction cascades leading from EGF stimulation through PLD to turning on this SOS protein, which ultimately leads to RAS activation. RAF1, another important kinase system embedded in the oncogenesis, is also activated directly by phosphatidic acid, which is generated by phospholipase D, and that results in activation of downstream mitogen activator, or MAP kinases. All of this, as you recall from previous talks I've given, and probably from your training, if you have training in biochemistry, you know that these are canonical pathways for altering signaling in cells, MAP kinases, SOS, these kinds of systems, all linked to this phosphatidic acid, you see. So PA can bind and activate even the mammalian target of rapamycin, or mTOR, which we've talked a lot about. This is an anabolic system which turns on basically cell proliferation because it's a protein kinase and it's involved in not only cell survival, but unfortunately, again, as I said, cancer. Likewise, sphingosine kinase, a topic we just covered in the last VeraMed lecture, has important roles in cell growth, calcium homeostasis, and cell movement. It's also regulated, this whole system, by phospholipase D and phosphatidic acid-dependent recruitment to intracellular membranes. So that enzyme itself is PLD-PA associated. So again, to give you a reminder, if you don't have this in the top of your head, which I'm sure most of you don't, because you're not lipid biochemists, but phospholipid metabolism is very, very elaborate and integrated in cell biology, particularly in normal physiology, but is also corrupted in pathophysiology that leads to disease states. Major players in membrane signaling involve the phosphatidylinositols. Remember that phosphatidylinositol can be phosphorylated multiple times. This phosphorylation, this particular molecular species, is highly reactive in signaling. This whole lipid can then be, of course, modified by phospholipase C, generating this sugar moiety, this trisphosphorylated sugar moiety, which is on its own right is a signaling molecule, and then diacylglycerol, which can turn on protein kinases type C. Diacylglycerol, of course, is a precursor to PA, but also you can make diacylglycerol from phosphohydrolase, PA phosphorylase enzyme. Phospholipase D, as we've been telling you, makes phosphatidic acid and choline, which can be recycled, for example, to make sphingomyelin. Phospholipase A2 is also associated with making the very important icosanoids from, for example, the omega-6 fatty acid arachidonic acid. It also regenerates lyso-PC. Lyso-PC can also generate lyso-PA. Lyso-PA, yes, indeed, can also form those phosphomonoesters. And lyso-PA is, again, really important in recruiting proteins to the membrane and activating them or deactivating them, depending. Remember, too, that once you make arachidonic acid, cyclooxygenases, lipoxygenases, and P450 monooxygenases will generate the whole family of prostaglandins and leukotrienes. Very important stuff. All right, time to put on the glasses. All right, so there are all kinds of highly complex interactions when you're talking about phospholipase D. There's transcriptional regulation, where growth factors can bind to receptor tyrosine kinases. There's your SOS, RAS pathway. There's protein kinase C, which can be induced by 4-ball myristate or by DAG, which is its normal ligand. At any rate, once RAS is turned on, you can turn on this kinase, which turns on AKT. AKT can then phosphorylate IK beta alpha, okay? Once that occurs, NF-kappa B can act as a transcription factor. All of that actually makes more PLD, which makes more phosphatidic acid, and more phosphatidic acid in this system with growth factor stimulation or diacylglycerol stimulation can lead to cell proliferation, okay? So that's, for example, a common mechanism in Ewig's sarcoma, okay? And that is, again, the main biofactor and the main molecule that drives this sarcoma is phospholipase D, okay? Phospholipase D2 in the particular sarcoma, and there's phospholipase D1 just in this continuous signal transduction cascade. Now, Wnt signaling is also associated with this. Wnt signaling can ultimately alter phospholipase D expression at the transcriptional level, and it can, when Wnt target genes are off, the genes that get shut off by Wnt signaling can be c-Myc and NOS2. That's nitric oxide synthase 2. Another very important signal transduction pathway in oncogenesis. And when Wnt is on, when Wnt's off, that's what happens. You shut down PLD. When Wnt is on, you make phospholipase D, that is your transcription of it, via the catenin pathway. Okay, you can see here all these players that are involved in creating this chromatin complex. Once you do this chromatin remodeling, you make phospholipase D. All the Wnt target genes are turned on because Wnt is turned on. It's signaling through this plasma membrane. And when that happens, you get tumor growth. So again, phospholipase D intimately involved in very core canonical pathways in the progression of many forms of cancer. So we're talking about phosphatidic acid, and we're talking about it being the product of phospholipase D. You see here how it plays a major role. There's phosphatidic acid playing a major role in all these pathways. There it is again for tumorigenesis. So that's why us biochemists are really interested in it. All right. Now, back to this BMC biology paper. Again, you have here this protonation of this phosphate monoester. Okay. There's an OH group directly associated with this. This is lysophosphatidic acid. So that 2 position of S-glycerol is free OH group, is an alcohol group there. Once you lose this proton, you can actually form a bond here, an intramolecular bond with that OH group. And that's really significant in whether or not this molecule can further react with an amino group. Okay? So hydrogen bonding increases the charge of both phosphatidic and lysophosphatidic acid. So that's really important. Stronger charge means going to be more interaction at the membrane. The phosphomonoester head group from lysophosphatidic acid, which is this molecule we're looking at here, forms this intramolecular hydrogen bond. Now in the protonated phosphomonoester of LPA, that's the one on the left, that's the protonated one, a proton is shared between the 2 hydroxyls, okay? Right. Hydrogen bonding, which are those green dashed lines there, right there, okay, between the SN2 glyceryl hydroxyl group and the phosphomonoester of LPA competes with that shared proton for oxygen electrons, because the oxygen electrons are ones that are competing for that proton, of course. That's going to facilitate dissociation of that proton, and it's going to lower the pKa. That's basically what's happening here, okay? That's what's going on here. Now, because you've lost that proton, you're no longer bound to this hydroxyl group here, okay? And now we're back showing you PA, but this could be lyso-PA too, but now we're looking at phosphatidic acid, which doesn't have that free alcohol group. Hydrogen bonding between the phosphomonoester of PA and the primary amine of the head group of PE, or a lysine residue, or an arginine residue of a protein. So it's showing you this could be PE or protein, you see. All of which is because of the lowering of that pKa because of the loss of that proton. This is where you get into intense then signal transduction, because now you've recruited either this phospholipid or this protein, which has this strong basic region. The basic region coming from arginine residues and lysine residues. Now binding directly to that phosphate monoester because of this formal -2 negative charge. Okay. So again, this is just looking at the same thing. Here you have the PA effector molecule bound here. This amino group's bound to this electronegative oxygen atoms from this phosphate monoester, right? You lower the pH and you get rid of the effector molecule, right? So this pKa of that monoester, phosphate monoester, then is dictating whether or not the effector molecule binds. This could be, again, a protein. Now it's lost, let's say, to the extracellular matrix, so that you no longer have the signal transduction with PA, all having to do with the pH. So see this molecule here, this phosphatidic acid, because of this unique interaction of these 2 oxygen residues in the phosphate monoester, and the loss of that second proton, is basically now the transducer of both measuring cellular pH and, because it measures cellular pH, the induction of the pKa loss of that proton and thus the effector molecule binding. So the PA embedded in the membrane acts as a pH biosensor, full stop. and acts as a signaling molecule by binding to effector compounds such as proteins or ligands, which will bind now directly because of the basic residues embedded in domains of the protein, the basic residues coming from arginine residues and lysine residues, for example, thus causing a signal transduction cascade directly through the PA moiety buried in the plasma membrane, or indeed in any intramembranous system. So, cellular pH, of course, is under tight metabolic control, and it has to be to modulate precisely the ionization of molecular species, okay? So we have to be careful not to see because if you alter the pH of the cell, you're going to ionize very important molecular species such as those amino acids found in proteins. So the level of protonation is going to ultimately determine the biological function. Basically, this is a pH gradient effect on the surface of the membrane and on the topological three-dimensional surface of the proteins which are associated with that membrane because they have ligand binding to that -2 formal charge because of the loss of the proton. So this establishes a lipid phosphomonoester center for PA to appropriate the biosensing of biological cellular pH. That's what's going on, appropriating it, apprehending it, making it so that the membrane can detect the pH of the cell and the circulating fluid. So the electrostatic hydrogen bond switch, this is called, and it's a mechanism which explains the ability of a phosphomonoester, such as that associated with phosphatidic acid bound to a membrane, to form hydrogen bonds with lipids and proteins, thus demonstrating the source of its pH sensing, as I've said, and the mechanism for pH-dependent effector binding. Okay, so proteins surveil then both the charged state of the phosphatidic acid as well as its loosening effect on the hydrophobic property of the membrane surface itself. So there's two things going on. Entire membrane fluid dynamics, that is the physical chemical fluid dynamics of the membrane alters when you make phosphatidic acid via PLD, production of PA from, say, PC embedded in the membrane. And thus it provides a mechanism for PA over other anionic lipids. PA is unique from lyso-PA because of that way that proton interacts. It's entirely possible that other membrane lipid phosphomonoesters, such as phosphatidylinositol phosphates, and ones we've recently talked about, ceramide 1 phosphate, the sphingolipid, and diacylglycerol pyrophosphate could also operate as pH biosensors. Okay, so we have all kinds of potential other membrane lipids. We have sphingolipids like sphingosine 1 phosphate, we have ceramide 1 phosphate, and we of course have PA. Okay. It seems that selection pressure directing biochemical evolution has enabled this electrostatic hydrogen bond switch in signal transduction. So selection pressure directed specifically on that biochemical moiety, that monoester. Again, here we take a look at a lipid moiety buried in the membrane, right? Here's a paper from the Journal of Biological Chemistry published in 2007, again showing you a very similar thing. Here is the canonical looking like a particular cone-shaped lipid in the membrane. This is going to be PA with its negative charge. Once you lose the second proton, you have two negative charges. It's going to pull a PA binding domain, such as arginine-rich regions or arginine-lysine-rich regions on proteins, and bind that phospholipid. See, it's sensing the pH, okay, because you're going to lose that proton when you alter the pH, right? So sensing the pH of the extracellular milieu, plus causing the signal transduction cascade, doing both jobs. So this is the model for increasing a negative charge of PA induced by positively charged amino acid residues. Okay, losing that proton there when it's reacting with that amino group from protein arginine lysine residues. The positive charge in hydrogen bond formation is shown for the primary amine of lysine, so that's what we're talking about here. The lower panel shows the interaction of positively charged protein domains with PA, as we've just covered. And remember that the molecular shape of the PA, that cone shape, the canonical cone shape, which allows for it to be selected from all other anionic phospholipids. Okay, so that cone shape, not all phospholipids with that unit charge, at negative charge, are going to have the same structure. PA does. So let's do a dielectric moment here. Not a dielectric, but a dielectric moment. Membrane phospholipids with phosphomonoester chemistry can sense the pH and induce effector signal transduction. That's a major statement we're making right now. That's an observation. That's evidence that I'm now verifying. This mechanism allows for very specific protein-lipid interactions in situ. In situ, what do I mean by that? That is Latin, of course, for right there. In situ, where is right there? It's in the monolayer of the outer leaflet of the membrane. Okay. So the fluctuation of cellular pH is common for signal transduction and pathogenesis and bioenergetics. Very common. And we're not talking about the change of like 2 pH units or 3 pHs. Never happens in living cells. The change of the only, remember, because this is a negative logarithm. So it's a power of 10. That's why pH has such a tremendous significant effect in biology, right? So all you have to do is change 1 pH unit and you drastically change signal transduction. So again, this suggests, it points to, okay, this is the conclusion of this dielectric moment, is that membrane lipids such as ceramide 1-phosphate and sphingosine 1-phosphate, topics of our last two, respectively, talks here at Vera Med Studios, they can be presumptive electrostatic hydrogen bond molecular switches along with the one we've described here and was described in the paper, the Langmuir paper, phosphatidic acid, because they're going to have the same monoester chemistry. So to show that, here's a paper that's published late in 2017 in my favorite journal of all, Journal of Biological Chemistry. And there's a citation. And look at the title of the talk, title of the paper. Phosphatidic acid binding polybasic domain is responsible for the differences in phosphoregulation of two proteins called lipins, 1 and 3. Okay, now, lipins are magnesium-dependent phosphatidic acid phosphatases. So they're stripping phosphate off of phosphatidic acid. And they catalyze actually the penultimate, that's the step before, of triacylglycerol synthesis. Why is that? With phosphatidic acid, remember I told you this was a really important like intermediate in lipid metabolism, not just in glycerol lipid metabolism, but in sphingolipid metabolism, and indeed, of course, in neutral lipids such as TAG. So when you take PA, you react it with a lipin, you make DAG, and DAG can be then acylated with one more fatty acid to make TAG. And this is the synthesis of the neutral lipid, for example, in the intramyocellular lipid droplet in muscle cells, but also, of course, in depot fat. Okay, so this is really important thinking about the dynamics of this in overall basal metabolism and in things like obese conditions. So this paper, they say they previously investigated the biochemistry of lipins 1 and 2, and they showed that dianionic, remember that's where we get that loss of that second proton, PA augments the activity of some of these lipins. And lipid binding, okay, associated with this lipin 1 activity is negatively regulated by phosphorylation of the PA, okay? So, and also of the lipins themselves. So you got multiple phosphorylation cascades here directing expression, or that is activity, the ultimate expression, I guess you could say, of a phosphatidic acid phosphatase, thus generating DAG, which of course, DAG itself, precursor to TAG, but also turns on protein kinase C. So a polybasic domain found in these proteins actually is composed of 9 lysine and arginine residues, and it's located within specific terminal domains of the lipin enzyme. And those are the site of the PA binding and sensing of PA electrostatic charge. The specific arrangement, number of those basic amino acids, lysines or arginines, of the PPD, that is the polybasic domain, vary among the lipins. So some of the lipins are turned on because of the presence of PA, and some of them are not because of the arrangement, the very specific arrangement of that PPD microdomain within the lipin protein. So that's why some lipins are turned on and some are not, because of their hard and fast association with membrane PA. Okay, so we're going to leave it at that. We left with a more recent paper. We started with a recent paper and we left with a recent paper. We went into the literature a little bit to verify those papers are connected. I want you to remember that this is Vera Med coming from the Pacific Northwest studios that we have. This is our email address for the web, and there's our website. Remember, we are scientists verifying published evidence in biomedical research and medical biosciences. There is my Facebook page where you can find everything about what I do in Vera Med, and that's me, and that's my specific email address when you want to talk to me about this particular lecture or any lecture previous, or indeed about what Vera Med can do for you. Thank you very much for your attention. So we're going to stop there and not continue on. That's for another talk. So hopefully you've enjoyed this talk, and I hope that you have a very pleasant week. This is actually Martin Luther King Day, so happy holidays for that very important holiday. And I just want to leave this by saying thank you again for your attention. Please contact me if you have any interest in describing further phosphatidic acid or any other topic that you have. Even though I'm just a lipid biochemist, I do have other functions in other biochemistry, pathology, pathophysiology, pharmacology, etc. Thank you very much once again for your attention. And as my sign off has been since quite a while now, goodbye for now.